Okay, so let's discuss Mendelian genetics. Okay, so the question is, how is the behavior of the chromosomes during meiosis related to the heredity? So in order to answer this question, we must first look back at the experiments of Gregor Mendel. The results of his garden experiments laid down the foundation of modern genetics. Okay, Mendel's discovery of principles of heredity. Gregor Mendel was a little information or history about his life. Gregor Mendel was an Augustinian monk in monastery of Brunn. Okay, in Austria-Hungarian Empire, now called as BRNO, in Czech Republic. He was interested in investigating how individual traits were inherited. Okay, he wanted to find out whether both parents contributed equally to the traits of the offspring. He also wanted to know if the traits present in the offsprings were produced by the blending of traits of the parents. Mendel chose the garden pea plant. It has a scientific name, Pisum sativum. Okay, for his experiments. Before he started his experiments, Mendel first produced pure breeding plants. Mendel allowed his pea plants to self-pollinate for many generations until all offsprings had the same features as the parents, generation after generation. So when he pure breeding plants, Mendel began cross-pollinating. Okay, cross-pollinating peas with contrasting traits. The pure breeding peas constituted the parental or P1 generation. All offspring of these crosses resembled one another. For example, when he crossed pea plants that produce round seeds with pea plants that produce wrinkled seeds, all offspring had round seeds. The offspring of the parental cross are called the first filial or F1 generation. In Mendel's experiments, the F1 generation are also called hybrids because they resulted from cross between two purebred plants with contrasting traits. For example, pea plants with round seeds with pea plants with wrinkled seeds. Okay. I'm going to show you table. Okay. The 7 pure breeding crosses that made based on 7 characters of the plant and the resulting traits of F1 generation. This is the results of Mendel's crosses between pure breeding pea plants. We're in character studied, and we have the traits from parents and their first filial generation. Excuse me. Hello, check, mic, check. Okay, when the plants from the F1 generation were crossed with each other or self-pollinated, the offspring F2 or second filial generation were of 2 types. For example, Mendel's counted 5,474 round seeds and 1,850 wrinkled seeds. In F2 generation, okay, so note that the round seeds were about 3 times as many as the wrinkled seeds. The ratio of round seeds to wrinkled seeds was 2.96 is to 1, or nearly 3 is to 1. Moreover, the wrinkled seeds or seed type did not appear in the F1 generation. For I'll be showing illustration that summarizes the results of Mendel second set of experiments. So here's the result of Mendel's cross between hybrid plants. Okay, so based on the results, of experiments, Mendel hypothesized that there was a factor in the plants which controlled the appearance of trait. Okay, these factors are what we call the genes today. Okay, since 2 alternative expressions of trait, let's say round or wrinkled seeds were possible, he hypothesized that the traits were controlled by a pair of genes now called alleles. Okay, Mendel's first hypothesis was in each organism there is a pair of factors that which controls the appearance of particular trait. Okay, Mendel noted that for each trait he studied, there is one trait that dominates the other. So based on the results of F1 generation, the trait for round seeds is the dominant trait. Excuse. The trait of wrinkled seeds, which did not appear in the F1 generation. Okay, again, the trait of wrinkled seeds, which did not appear in the F1 generation, is called recessive. recessive trait. Its appearance was either prevented or hidden by a dominant trait. This is known as the principle of dominance. The dominant trait dominates. The dominant trait dominates or prevents expression of recessive trait. Today, dominant and recessive traits are represented by a capital and small letter, respectively. Let's say A, capital A, and A, small a. Okay, a dominant trait like a round seed, for example, is represented by R, big letter R. And recessive trait like wrinkled seed is represented by small letter r. Okay? Since genes occur in pair in pure breeding round seed plant, is symbolized as R, as RR, double R, which are capital, and a pure breeding wrinkled seed or seed pea plant as RR, small letter R. Okay, which are dominant and recessive traits of peas studied by Mendel. Okay, let us move. So here is the dominant and recessive traits in Pisum sativum or garden pea plant, which shows the characters studied. Dominant traits, here are the dominant traits: round, yellow colored, inflated, green, axial, long, and then recessive trait. Okay, for the other one, law of segregation. Hello, check mic, check. Sorry. So before Mendel's time, it was believed that all traits become mixed when they are transmitted from one generation, okay, to the other generation, as red and blue paints mix to give a violet. Now let me write that first. Okay, violet color. However, when Mendel crossed pure breeding pea plants, the pea plants did not produce offspring with blended intermediate traits. In Mendel's experiment, the pure breeding parent plant had two identical genes for a trait. Round seed. Let's say round seed. Okay. And for the wrinkled seed. Okay, Mendel hypothesized that the pair of genes segregate or separate from each other during gamete formation. This is known as law of segregation. Okay. So for another is genes and gametes. Following Mendel's reasoning, a purebred or purebred round-seeded parent plant has an allelic combination of genotype of RR, double R, which are capital letters, while a pure breed wrinkled seed parent plant has a genotype of small letter R's. Okay. Individuals that are pure breeding for a particular character therefore have identical alleles. These individuals have homozygous. genotype. For seed shape, a homozygous dominant individual will produce round seed, while a homozygous recessive individual will produce wrinkled seeds. Okay? Homozygous genotype. Okay, next. Mendel's law of segregation states that the genes of a pair, for example, RR here, separate during gamete formation. So one gene goes to another or to one gamete, while the other gene goes to another gamete. Since the gene of the pair are alike, all gamete produced by the homozygous parent will be alike. So all have gene R. Okay. The same is true for the other parent. All of its gametes have gene small r. Okay, let me show you their traits. Round and wrinkled. Okay, big letter R's and small letter R's. Okay, that will have separate R's, which are called gametes, and small letter R is also gametes. Okay. This is a gamete formation for pure breeding round and wrinkled seed, okay, bearing pea plants. Now recall what we have learned in meiosis during anaphase 1 of meiosis 1. The chromosome pairs separate and move the opposite poles. Now remember that A trait is governed by a pair of alleles. So each allele is found in a chromosome of a chromosome pair. In other words, the chromosome serves as a vehicle for these alleles. So when the chromosomes segregate during anaphase 1, the alleles they carry also segregate. This becomes chromosomal basis for Mendel's first law. Okay. We're in chromosomal basis of Mendel's. Okay, I'll show you the basis of Mendel's law of segregation. Ganito na lang. Ipapakita ko na lang sa'yo. I will not draw anymore. So here's the chromosomal basis of Mendel's law of segregation. Okay. At fertilization, when the gametes formed during gametogenesis by RR, or traits round and wrinkled, plants unite. So all zygotes will have the genotype RR, which are small and big. Okay. Thus, all the F1 or first filial generation plants will have the genotype of R, big R and small r. An individual with contrasting allele. Okay, a dominant and recessive allele for a particular character is said to have a heterozygous. Okay, this is now heterozygous. Genotype. However, the RR individuals will still produce round seeds because of the presence of dominant allele. This will be just a round as all of the seeds produced by RR parents. The expression of the genotype of an individual for a particular character is referred as phenotype. Okay, so expression of genotype of an individual for a particular character is referred to as phenotype. Okay. So let us crossbreed the round and wrinkled. Okay, so big R and small r. Okay, in fertilization, process of fertilization. Okay, gametes. Will produce round seed. Okay, so this is the results of crossing pure breeding round and wrinkled. Okay, so this is the first filial generation. Excuse. Knowing the genotype. So when you observe pea plant, you can easily see pea plant yellow pods or actual flower. By simply looking at the plant, you can immediately tell a plant's phenotype. However, you cannot always tell its genotype. For instance, a plant with a wrinkled seed certainly has a RR genotype. But what about the plant with the dominant trait, round seed? Its genotype could either be R, big RR, or big R, or combination of big R and small r, which are dominant here, and then are homozygous and heterozygous. There is, however, a way of knowing whether a plant is homozygous or heterozygous for a given character. So Mendel's applied two techniques in determining genotype of a pea plant. These are self-fertilization and test cross technique. Excuse. Okay, for self-fertilization technique. Okay, when self-fertilization fertilize homozygotes, always produce pure breeding plants. Thus, round seeded pea plants or peas will breed only round seeded peas. What about heterozygotes? Again, heterozygous round F1 or first filial generation peas produce round and wrinkled seeds in the ratio of 3 round seeds and one wrinkled seed. This ratio is called as phenotypic ratio. Okay, self-fertilization enables us to determine whether an individual is homozygous or heterozygous. For a given trait by observing the phenotypes, okay, of its offspring. Homozygotes are pure breeding. Heterozygotes produce two kinds of offspring. 3 out of 4 offspring show the dominant traits, and 1 out of 4 excessive the recessive trait. Okay, the next one is test cross technique. Okay, test cross involves a cross between the recessive parental type, okay, which are wrinkled small r's in our sample cross. The individual with the dominant trait, okay, which are here, but with unknown genotype. Okay, you will observe in here that homozygotes or homozygote dominant, okay, dominants when crossed with recessive produce only one kind of offspring, and all exhibits the dominant trait. Okay, heterozygotes, on the other hand, when crossed with recessive, produce two kinds of offspring. About one half of the offspring show the dominant trait, while the other half exhibit the recessive trait. So let us see. Let us have the round and wrinkled. Okay, so let's have their symbol. Okay, so wrinkled, let's crossbreed them. Small r. Let us have box here. Big letter R from the left, which are round gametes. Okay, so when you crossbreed them, it will produce R, big R, and same with the other one. Okay, they are all round because of the dominant trait. Okay, first filial generation is all round. The other one is wrinkled gamete, same with this. Which has the same big R. Okay, combining them, big R and small r, big R, small r, big R, small r. So for the first filial generation is 1/2 round and 1/2 wrinkled. Okay, determining its phenotype. Okay, this is what we call the test cross technique. Okay, so let us move. Probability. Okay, when we talk about the probability, the chance plays a role in it in the inheritance of trait. During fertilization, the union of male and of male gametes with the gene R, big R, and the female gamete with the gene small r, for instance, okay, happens by chance during meiosis. The assortment of different chromosomes also occurs by chance. Okay, we can also illustrate the role of a chance in inheritance of the trait. Okay, back. Okay, so let us have diagramming a cross. So Mendel's crosses can be recorded in a chart called Bonnet Square. What the? Okay, so the Bonnet Square help us to predict the outcome of a given cross. It allows us to determine the possible combination of gene in a cross. So the use of the Bonnet Square can be illustrated by solving an actual problem, okay, like determining expected result when two hybrid round seeded, okay, peas are crossed from the when they are crossed. Okay, you know that the expected phenotypic ratio from this cross is 3:1. So we'll be determining the expected genotype of the offspring. Okay, so since the zygote of the genotype, I have a question. So how many kinds of genotypes are possible in a cross, and what are these genotypes? Again, how many kinds of genotypes are possible in a cross, and what are these genotypes? Okay, let's continue. Since zygote with a genotype heterozygous alleles, okay, big R and small r, is shown in twice in the Bonnet Square. Okay, so the chance of probability that a zygote will have a RR genotype is 2 out of 4. Okay, 2 out of 4, or we can have 2/4 or 50%. Okay, pwede naman natin gamitin to or ito. Kahit ano naman dyan. 1 out of 4 or 1/4 or 25% will have the genotype RR, big R, double R's. So while 1 out of 4 will have the genotype of RR, again, small r naman. Small letter R's. Therefore, the genotypic ratio in the given cross is one of RR homozygous. Okay, round seed. And two RRs, which are big and small, combination of big and small, heterozygous. Round seed. Okay, to 1 R, okay, homozygous, wrinkled seed. Okay, so the genotypic ratio is simply written as 1 is to 1 is to 1 is to 2 is to 1. Okay, this is this 1 is to 2 is to 1 ratio is what you should have obtained as your ratio. And activity number 3. Okay. Okay, let us have an example of round seed. Okay, male and female. So for male and female, we have heterozygous, parehas, which are already combined. Okay, we have male and female. Okay, so the gametes of female here are heterozygous to and for male here okay, combining both big R will have homozygous round. Okay, then this one and then this one, small, big and small. Okay, when we are combining them, make sure that we are writing the first big letter, dominant letter, okay, followed by small letter R. Even though small letter R is placed first before, or written first before big R here, or on the left side. When it is combined, when they are combined, you make sure that you write big R first, followed by small r. Okay, same with the other one. Okay, so the genotypic ratio of 1/4. Genotypic ratio is 1/4 RR, big letter R, and then 1/2. Why is it, sir? We only have 1/4 RR o big letter R's. Okay, kasi dahil sa apat to, 1 out of 4 we got 1. Okay, 1/4 RR genotypic ratio. Then 1/2, dahil apat to, kalahati niyan nakuha nito. Okay, heterozygous round. Okay, so R, big R and small letter R. Next ay 1/4 ulit, pero homozygous wrinkled. I'm sorry for this, nagkamali ako rito, wala ako sa. Okay, so double R combining this one, okay, will produce small letter R's. Okay, so 1/4 of homozygous wrinkled produce 1/4 only. So 1 and 2 and 1/4 and another one For phenotypic ratio, let us have phenotypic ratio. What the? Okay, is 3/4 round. Okay, when we are talking about phenotypic ratio, we are pertaining to its physical appearance. Okay, 3/4 round. Why is it 3/4, sir? Kasi tatlo ang round. Isang homozygous round and dalawang heterozygous round. Bakit round pa rin to, sir? Kasi diba they are dominated by dominant trait, which is the round. Okay, 3/4 round and then 1/4 wrinkled. Bakit wrinkled, sir? Homozygous wrinkled here. Okay, kakaintindihan po ba? Okay. So let us move excuse cross, okay, with 2 factors tayo, which is what we call dihybrid. Okay, Mendel also studied the inheritance of 2 pairs of factors in an individual. A cross between individuals that involve 2 heterozygous pairs of genes is called dihybrid cross. So Mendel crossed a plant with genotype excuse me, they're all big, so round yellow seeds, and they plant with a genotype of small r's and small y's, which are wrinkled and green. So round and yellow, wrinkled and green. Small y is green. Okay. Excuse. So the F1 piece exhibited the dominant traits of heterozygous phenotype, which is this. Okay, heterozygous round yellow. Okay, assuming that Mendel's law of segregation, each pair will segregate during the formation of gametes. So each gamete will therefore receive 1 allele from each pair. So if the pairs of alleles are found on different chromosomes, then each dihybrid will produce 4 different types of gametes with 1/4 probability. 1/4 probability. Okay, so let us show the gametes produced by dihybrid genotype. Okay, we have R, big R and small r, big Y and small y. Okay. Okay, we have 1/4, 1/4, 1/4, 1/4. So these are the gametes produced by dihybrid genotype. For the self-fertilization, okay, so self-fertilization of F1. copies produce 4 phenotypes. Okay, the phenotypes with a ratio of 9, 3, 3, 1. Round yellow, round green, wrinkled yellow, and wrinkled green. Okay, so there is an activity that I'm giving you, wherein you will be able to use Bonnet Square, okay, solving dihybrid crosses. Okay, so given the cross of heterozygous round yellow, and same with this round yellow, okay, using Bonnet Square. Okay, Bonnet Square is the box. Okay, that we use to combine with letters. Okay, wow. Okay, hello, check mic. Okay, we'll be rewriting the same with the same letters here. Big RY, big R, small y, small r, and big Y, and homozygous round green. Okay, when we describe big R, this is a round, dominant, round, wrinkled, and wrinkled, yellow, big Y, small y, green, yellow, green, parang ganon. Okay, combining this, we'll have double R, big R, and double big Y. Okay, then combining this also give us double big R. Then when we're combining different letters, we make sure that big letter right is written first before small. Okay, another one is R, big R, and small r, and then big Y, double big Y. Okay, next big R, small r, and for Y, big Y and small y. Okay, that is how we solve this. Okay, okay, so we'll be getting the genotypic ratio, and phenotypic ratio. Okay, so we solved using Bonnet Square. Okay, so I hope you like this, and if you understand the lesson and you find this useful or helpful, you may like and subscribe. But before that, I'll be continuing the discussion for non-Mendelian. Okay. There is what we call non-Mendelian pattern or patterns of inheritance. Okay, so all of the traits controlled by dominant and recessive. Recessive gene. Okay, what about traits that have more than 2 phenotypes? Do they follow Mendelian patterns of inheritance? So this is what we call incomplete dominance. Okay, when a pure red flowered 4 o'clock plant is crossed with a pure white flowered 4 o'clock plant, the offspring will produce neither red nor white flowers. Instead, all flowers will be pink. In other words, the offspring will have phenotype intermediate between phenotype both parents. Such a phenomenon is called incomplete dominance. This means neither of the two alleles is completely dominant, and heterozygous will be new phenotype. Therefore, genotypic ratio is also becomes the phenotypic ratio. Okay. Next, codominance. Okay, codominance. Another pattern of inheritance is this one. So this result is when one allele is not dominant, one allele is not dominant over the other. Okay, resulting heterozygous heterozygotes exhibit the traits of both parents. Okay, one example of codominance is MN blood typing. In humans. So, on the surface of our red blood cell are proteins. Okay, so bound to sugar molecules forming complexes called antigen. Okay, so one group of antigen are controlled by a pair of alleles. So one group. Okay, so one group of antigen is controlled or are controlled by a pair of allele, which is the LM and LN. The pairing of these alleles will determine the blood type of an individual, and there are 3: M, MN, and N. This is a table that summarizes the genotypes and phenotypes of MN blood typing. Okay. So let us have the note. Okay, that in heterozygote condition, both LM and LN alleles are expressed in that the red blood cells will have the M and N antigens. Okay, like in incomplete dominance, the genotypic ratio also, codominance, also becomes the phenotypic ratio. Okay, for multiple alleles, sometimes even if only two alleles control a trait, there may be more than two types of allele available. Okay, this will also lead to a more than two phenotypes expressed. Another blood group system in humans, the ABO system, is an example of a character governed by multiple alleles. Three alleles are responsible for this blood system. Okay, we have this one. Okay, the ABO blood type is determined by the presence or absence of two antigens, A and B. Okay, A and B allele or allele I does not code for an antigen. There are four possible blood types as shown in this table. Okay, so this is a human ABO blood types and their phenotypes. Okay, that's it. We'll continue this discussion next time. I'll be uploading next topic, which is the continuation of the topic for genetics. Okay, thank you.